Hello, friends. A material which has some magnetic properties will develop a magnetic field around them so that it can either attract or repel another magnetic materials. Take iron fillings for an example. If you bring a magnet close to this iron fillings will get attracted towards the magnet. Our red blood cells contain a lot of hemoglobin, which in turn contains iron. Will the magnets attract these iron present in our blood? Let us explore this in this video. I'm your assistant. You are watching Students of Chemistry. If the magnets attract the iron in our blood, then I must say we all are in a grave danger. But good to us, the magnets doesn't attract the iron in our blood. So we must find it why. While researching, we will always end up with this molecule called hemoglobin. Our red blood cells contain a lot of hemoglobin, which I already said. This hemoglobin contains a protein called heme, which contains the hero of this subject called iron. This hemoglobin can either be deoxygenated or oxygenated, which means it can either be attached to oxygen or it doesn't attach to oxygen. These are called as oxygenated hemoglobin and deoxygenated hemoglobin. One must know that in both these forms, iron is present in the +2 oxidation state. So its electronic configuration will be 3d6 4s0. These 6 electrons will occupy the d orbitals in this manner. We can see that the deoxygenated hemoglobin without oxygen, iron has 4 unpaired electrons, so making the molecule paramagnetic. The paramagnetic materials are those which will be strongly attracted, or I must say feebly attracted towards the magnetic field. When the oxygen comes into the play, oxygen has an electronic configuration of 2s2 2p4, and this is the manner in which the electrons will be filled in the p orbital of oxygen. So these 4 valence electrons will covalently bond with the iron 2 plus to pair up all the 4 unpaired electrons. Not a coordinate bond, it is a covalent bond. So this is the oxygenated hemoglobin, which contains all paired up electrons, making the molecule diamagnetic, or in other words, it will be feebly repelled by the applied magnetic field. But one must note that diamagnetic character is a weak magnetic character. So if a molecule contains paramagnetic and diamagnetic character, the paramagnetic character prevails. Thus, we are again stopped at a point where we must say blood is attracted by the magnetic field. As the hemoglobin's job is to deliver oxygen to all the parts of the body, it will be always found in the oxygenated form, making the molecule more diamagnetic, making the diamagnetic character more dominant compared to the paramagnetic character. It is not stopped with this. The H2O molecule contains 2 protons which has 1 unpaired electron each, and oxygen contains 2 unpaired electrons. These unpaired electrons will bond together to form 2 covalent bonds, making the molecule diamagnetic, which is again prevailing the diamagnetic character, so that our blood is totally showing diamagnetic character to the magnetic field compared to the paramagnetic character. So thanks to water, we are saved and we are not attracted by the magnetic field. So if my blood contains diamagnetic character, then I must say my blood will be repelled to some extent in the external magnetic field. You may not experience with small magnets, but when you're placed in between super magnets containing creating a large magnetic field, you may experience some repulsion. Take your blood sample and stick it to some thermocol or something and make it float in the water. If you bring a magnet close to this blood sample, you may see that this blood ripples and floats far away in the water compared to the original position. This is happening because our blood is actually repelled to the external magnetic field. So if there is any tool to measure the paramagnetic and diamagnetic characters separately? Yes, there is a popular tool named magnetic resonance imaging, or MRI. In hospitals, these tools are used to image our parts of the body. This MRI consists of a strong magnets which differentiates the paramagnetic character and diamagnetic character of the blood, so that giving the images of the parts which we needed. This technique is called as BOLD, or blood oxygen level dependent. If I Google the word BOLD, or blood oxygen dependent level, in the web and choose the Wikipedia's page, it says that blood oxygen level dependent is used to image the different areas of brain or some other organs by differentiating the oxygenated hemoglobin's magnetic character and deoxygenated hemoglobin's magnetic character. Thank you for watching this video. If you like this video, please like, share and comment. Don't forget to subscribe to our channel. Click the bell icon. Thank you so much.